This part of the lecture, we're going to cover a few more fundamentals of water flux or movement in trees, including diffusion versus bulk flow, the cohesion-tension hypothesis, and then soil-water relations, the movement of water in roots, and the storage of water in stems and branches. So wanted to start talking about diffusion versus bulk flow. That's sort of two main mechanisms by which water might move around in a tree. Generally, diffusion implies movement of water. Could be other substances, but it's usually across a membrane. And it's down a gradient in total water potential. So when we talked about water potential, recall psi W is the total water potential, which is the sum of pressure and osmotic potentials and gravitational. And diffusion would just be moving across a semi-permeable membrane down a psi W gradient from less negative to more negative. Whereas bulk flow refers to flow of water through a conduit. And I'm going to use the term conduit to just generically refer to sort of a pipe. In the xylem, when I say conduit, generally I'm talking about vessels or tracheids, but it could also include an aquaporin in a cell membrane that I'll talk about in a minute. But it's just flow of water through a conduit down a gradient. In this case, it's usually down a gradient in pressure. So usually in that pipe, whether it's a vessel element or a tracheid, there's a pressure potential gradient, sort of like in a drinking straw. And I'll give you some examples in a second. Now these two mechanisms, diffusion and bulk flow, essentially operate at different scales. Diffusion is really effective across very small distances. In other words, from one cell to an adjacent cell, for instance. Diffusion is an effective mechanism of transport of water from one cell to the next. But diffusion, we know diffusion can't be the mechanism of water transport from the roots of a tree to the crown of the tree because it's too slow. Diffusion would take years. For water to move from root to leaf, if diffusion was the mechanism. So bulk flow is really the mechanism for long distance water transport. When we talk about water movement in the xylem, we're talking about bulk flow in tracheids or vessel elements. That's how the water is moving those long distances. Here's an example of bulk flow that we're all familiar with, drinking out of a drinking straw. So we've got a glass here with water in it, and there's a straw immersed in it, and we put our mouth around this part of the straw. This end of the straw is at atmospheric pressure, essentially. And when we put the straw into our mouth, by sucking on the straw, we create pressure in our mouth that's slightly less than atmospheric pressure. And when that happens, that pressure difference, so this might be P1 and P2, where P1 is greater than P2. When that happens, there's a pressure gradient, and water moves by bulk flow down that pressure gradient. That's the same mechanism by which water moves in the xylem, except in that case, generally, the P1 and P2 may be both negative. It's just that P2 is more negative than P1, but there's still a pressure gradient. So this is the simple way of thinking about it, is just the drinking straw and us generating a pressure gradient. Now, of course, this is similar to, for instance, the vacuum pump that we spoke about. Vacuum pumps work the same way. They generate P2, a pressure that's less than atmospheric, and that pulls water up the pipe. And there's a limit to how long that pipe can be, because P2 cannot be any less than the vapor pressure of water. And that's the difference in plant and tree xylem, is that P2 and sometimes P1 are negative. They're much lower than the vapor pressure of water. And that's sort of the special thing. But functionally, we're always just talking about pressure differences and the bulk flow of water down that pressure difference. So sometimes keeping the drinking straw in mind can be helpful. Here's a diagram of a cell membrane. So this is just the cell membrane here. And it's showing those two mechanisms of water transport, where this is outside the cell, this is the cytoplasm, and water can move by diffusion. That's what's being shown here. That's just diffusion down a water potential gradient from outside to inside. But water can also move by bulk flow through an aquaporin. And an aquaporin is just a protein embedded in the cell membrane that can open and act as a channel through which bulk flow can occur. Aquaporins were first discovered in animal cell membranes, I think probably back in the 80s, maybe the 90s. But they were subsequently discovered to exist in plants as well. So plants have aquaporins as well. And aquaporins just provide a much more efficient mechanism for water to move into or out of cells than diffusion. So bulk flow can also occur across cell membranes as well. Now, a really important concept for modeling and understanding water flow in plants is this Ohm's Law analogy. Now, Ohm's Law is an electrical concept, but Ohm's Law basically says that the current in a circuit equals a voltage difference divided by the resistance in that circuit. So the current flow through a circuit is a function of the voltage divided by the resistance. So in any particular wire, for instance, given the same voltage across that wire, the current will vary depending on the resistance of the wire. So this is just a restatement of Ohm's law, where flux is the amount of water flow. The potential difference is just the delta water potential difference. It might be pressure potential, it might be total water potential difference between 2 points. That might be root to leaf. That might be branch to leaf. But just sort of defining those endpoints in what the water potential difference is. So again, remember, water always flows from less negative to more negative. This is just the difference in water potential between those 2 points. And then resistance is just what it says. It's the resistance to water flow in the xylem between those 2 points. Okay, so it allows us to analyze movement of water in plants. This is also used in other systems. Sometimes it's called Fick's law. To analyze, for instance, movement of water in soils. But they all have similar concepts in terms of looking at a flux, a potential difference, and a resistance. You know, thinking about this in terms of pipes, I'm coming back to pipes or conduits. These might be xylem elements, for instance, where the flux is, so the amount of water moving through pipe. You could even think of this as a garden hose, for instance, is another way to think about it, where there's a pressure on one side of the conduit and a pressure on the other side of the conduit. So that potential difference is just P2 minus P1. So let's say we have these two situations. Whoops. Where we've got a pressure here, pressure there. And let's say these are equivalent, where P1 in the first case is the same as P1 in the second case, P2 in the first case is the same as P2 in the second case. So that potential difference is the same in these two situations. In which situation will the flux be the greatest? Well, it's sort of intuitive that we'll get greater flux in this situation. Given the same potential difference, you'll get greater flux in the bigger pipe because the resistance, let's say this is resistance 1, this is resistance 2, the resistance of that top pipe is greater than the resistance in the second pipe because it's smaller. Same thing works in xylem. Some pipes are narrower than others, and that affects the resistance and therefore affects both the differences in water potential gradients that are developed as well as the rate at which water can flow through those pipes. You can think of this as a garden hose or a drinking straw. At a given pressure difference, you'll get more water flow through a big pipe or big hose or big straw than you will through a little straw. Similarly, to move the same amount of water, for flux to be the same through the top pipe versus the bottom pipe, you have to generate a stronger potential difference, a stronger pulling force, or have a bigger difference between P1 and P2. So just keep in mind those interrelationships between potential differences, resistances, and flux. So for instance, if we wanted to analyze water flow through a tree, we can use these kinds of relationships. So for instance, we have ways that we can measure sap flow through the stem of a tree. That just equals the flux. And if we can measure the water potential of the roots and the water potential of the leaves, we know what flux is. So that would just be sap flow equals psi root minus psi leaf divided by resistance. So this term and this term are known. We measured those. So we can solve for this term, for the resistance term, and that will give us an estimate of the total resistance of all the xylem between the root surface and the leaf. That's an interesting measure of the hydraulics of that system. Similarly, we might measure water potential here and here, or measure water potential across a branch from here to here, and solve for and measure flux, measure water potential, and solve for resistance, and that will give us the resistance of the xylem in that segment. So this Ohm's law analogy is used a lot in plant physiology to understand flux and resistances in different parts of the pathway. This is another really important concept in water transport in vascular plants, and that's the cohesion-tension theory. This is the consensus theory for how water transport occurs in vascular plants. Back in the 19th century, there were a lot of working hypotheses for how water transport in xylem might work. And in the early 20th century, cohesion tension sort of arose as the leading theory or hypothesis. And I think has settled into the role of theory, where there's a lot of data supporting this mechanism. As I mentioned before, occasionally there are challenges to the cohesion tension hypothesis or theory, sometimes because people have a hard time accepting the existence of liquid water in a metastable or negative pressure state in xylem, which is necessary for cohesion tension to work. But for the most part, physiologists working in water relations agree that this is how it happens. So there's a few tenets of the cohesion tension theory that also explain how it works. And one is that there's a continuous column of water from the roots to point of evaporation in the leaves. In other words, there's a continuous and uninterrupted chain of water from the root surface all the way through the roots, through the xylem and the stem, through branches, into the leaf veins, and into the cell walls of mesophyll cells where the evaporation into the intercellular spaces of leaves actually occurs. That's a continuous column or rope of water. Another important point is that there is cohesion between water molecules. Since water is polar. And what I mean by that is the molecular structure of water looks something like this, where this is the oxygen atom, and these are the two hydrogen atoms. So this molecule is polar in that there's a slight positive charge on this side of the molecule and a slight negative charge on this side of the molecule, because the positive valences of hydrogen are over on one side and the negative valence of oxygen's the other side. And what that does is that means that water molecules tend to stick to each other like this. So that net positive on one side is attracted to the net negative of the other side of the next molecule, and they tend to stick together. That sticking together is called cohesion, and it's pretty strong. Water is quite a polar molecule. That's also what makes water a really good solute. It's because that polar nature can pull other molecules apart and dissolve them. It also means we have surface tension in water, which is a special attribute of water, and that's because water is polar. Okay, so cohesion occurs, water molecules stick together. Third, there is tension or negative pressure develops due to evaporation of water from leaf mesophyll cell walls. And I'll elaborate on this in a minute. So in other words, that evaporation generates a pulling force. Okay? That pulling force pulls the water column upward. Okay, so there's a continuous column of water. The molecules in that water column stick together. As water evaporates from the cell walls, that generates a pulling force that pulls the water column up the tree. Okay? One special note, water flow is not due to capillary forces in xylem conduits. It was thought for some time that perhaps capillary forces were what caused the rise in water in sap. So when I say that, what I mean is that if you place a narrow tube into water, because of its polar nature, will stick to the edges of that container and flow upward in that tube a little bit. That's called capillary rise. And it's just due to the attraction of those polar water molecules to the walls of the tube. If you get your finger pricked for a blood test, sometimes they'll take a little glass capillary tube and touch it to the drop of blood on your finger, and the blood runs up that capillary tube. That's due to capillary forces. Capillary forces are a function of the narrowness of the container. So the more narrow the container, the stronger that pulling force due to capillary forces. But xylem conduits are only narrow enough to pull water upward about a meter. Okay, so capillary forces could in, for instance, tracheids, could account for pulling water up maybe a meter. Okay, so we know that this flow of water through a xylem of a tall tree is not due to capillary forces in the xylem. So tracheids, the narrowest tubes, for instance, are not narrow enough to generate sufficient capillary force to cause that. Okay, so I just told you that capillary forces in xylem conduits are not the source of the pull that moves water by cohesion tension. I don't want to confuse you, but capillary forces are the source of pull when we're talking about the capillary forces in mesophyll cell walls. So capillary forces in mesophyll, so I'll say leaf mesophyll cell walls do generate the pulling force that drives cohesion tension. And here's how it works. This is a great diagram from the Graham et al. Plant Phys book. So we're looking at, this is a cross section of a leaf, and we're going to zoom in really close. So here's a cross section of the leaf. Okay, we're going to zoom in on some mesophyll cells. Okay, so that's the zoom. Each of these is a mesophyll cell with the intercellular spaces of the leaf. Okay, now we're going to zoom in even further just on the cell wall of one mesophyll cell. Okay, this is the cell wall right here of the mesophyll cell. And it should look familiar. I've showed you images of cell walls, which are just a bunch of cellulose microfibrils laid down across each other to make that cell wall. Okay, now we're zooming in even more. Here we're zooming in and showing just two cellulose microfibrils where they cross in that cell wall. Those two cellulose microfibrils make a little pocket. Basically, you can think of this as a little container, almost like a little tube where water is. So when water evaporates from a mesophyll cell into the intercellular space, it's basically evaporating from the cell wall from in between those cellulose microfibrils. As that water evaporates from those cellulose microfibrils, it forms a meniscus. So this curvature between the cellulose microfibrils is a meniscus that's caused by the clinging of those water molecules to the cellulose microfibrils. Just like water in a capillary tube clings to the walls of that tube, the water clings to the sides of those cellulose microfibrils. And the more the water evaporates, the steeper that curve becomes. So as more water evaporates, the radius of curvature of that water, of that meniscus, becomes steeper, and the pulling force becomes stronger. This meniscus generates the capillary pulling force. And because this is such a small container, it's like a tiny, tiny tube. Orders of magnitude, these distances are orders of magnitude smaller than, for instance, the smallest trachea. Cellulose microfibrils are microscopic, right? They're at the molecular level. So these are very small tubes, and that means that meniscus can generate very strong pulling force. This is looking at different radius of curvature. This is a huge pulling force. So these pulling forces can be very large. So this is the point. That point of evaporation is where that pulling force gets generated. So it's really important to distinguish this capillary force that's basically between cellulose microfibrils in the mesophyll cell wall, to distinguish that from any capillary forces that exist in, for instance, xylem conduits. They're different. This is the place where the pulling force for cohesion tension is generated.